Hello, class. I wanted to go over the week 7 video lab presentation on DNA extraction. And so I want to go over a little bit about what you'll be doing in the lab and show you some things. So first of all, I want to go over this portion, the materials. And so to go over the materials, I do want to show you. And so I'm going to stop sharing for a minute. Look at my video. Okay, so that you can actually see what I have in front of me. Okay, and so make that a little bit smaller so I can tell you what you got. All right, so you're going to use these plastic cups. And so these are the little small cups that came in your kit. On this one, I drew a line here to show what a tablespoon looks like, and I will tell you why in a moment. All right, so the two plastic cups, and then a couple of these droppers, okay? Two plastic droppers. You need some detergent, doesn't have to be Dawn, but any liquid detergent works. And then you need your salt, okay? And contact lens solution or meat tenderizer. Well, I wear contacts, so I had the contact lens solution. But meat tenderizer works as well. Any contact lens solution is perfect. You need some isopropyl alcohol, but it does need to be 91%. Okay. So I got this at Walmart. Just to give you a heads up, I got this at Walmart, but I'm sure they have it at any drugstore, CVS, Walgreens, any other place where you can get pharmaceuticals. You can find 91% isopropyl alcohol. You need a baking dish. Now, the baking dish, basically, what the baking dish is for is because you have to heat these up. So you're sitting these in some hot water. And so you see how small these cups are. You don't need a big baking dish. Okay, so what I took is I actually have my water in here, but it's just a bowl. Okay, just a large bowl. Just something that your cups can sit down in and heat up, okay? And you're gonna have hot water in there. So it has to be something that can take hot water. Then you need some toothpicks, okay? You got a toothpicks, and you don't need a lot of them. I did the experiment with 4 toothpicks, okay? It says you need approximately 15, you don't. You need about 4. All right, and you need coffee filter. So coffee filters, or you can use a thick paper towel. Okay, either one will work perfect. And then saliva. This is kind of gross, but you gotta spit into the cup. All right, so you need your own saliva and need some strawberries. Okay, you just need one small strawberry. Okay, so you can enjoy the rest and just eat them. All right, so I'm gonna go back to sharing the screen. Okay. All right. So what you're going to do is you are looking at extracting the DNA from the strawberry and from your own saliva. So one thing I want to go over the instructions, and then I want to go over just some highlights of the procedure and then some tips that I found when I actually did the experiment. So DNA extraction requires that a sample of living material be subjected to 2 main steps, lysis and precipitation. Because the cellular structure varies a little depending on the cell type and organism, the materials and specific steps taken to extract DNA will vary just a little bit among your samples. Additionally, a cleaning step to remove the protein and other cellular debris may be included. All right, so the procedure, okay, read through, excuse me, the lysis portion because you are extracting animal cells, which is your human cells, that's the saliva, and plant cells, which is the strawberry, okay? And so you're collecting your saliva sample in a plastic cup from the lab kit, which I just showed you. And this is done by spitting into the cup. The saliva contains cells from your cheeks and mouth lining. Your goal is to collect saliva to fill approximately half the cup or approximately 1 tablespoon. Okay, so remember that line on the cup that I showed you? Trying to find it. There it is. The line on the cup. I don't know if you can see it. There we go. This is a tablespoon. Okay, so what I did is I measured out a tablespoon of just water, poured it in here, and then drew a line with the Sharpie. That's to let you know how much you need to spit into here. Okay, so you're going to spit till you get to the line. You don't have to do a half a cup. As you can see from the picture of the line, oh no, it's hard to see that line. I'll make it a little bit darker so you can see it. Okay, hopefully that's better. Okay, there we go. Okay, as you can see from the line, there's not much that you're spitting into this cup, okay? So make sure you go to that line though, because you do need about that much in order to get a good sampling of your DNA, okay? And so you're gonna spit into the cup, and then on your other cup, you're gonna put in the strawberry and you're gonna mash it up really good, okay? And so a small strawberry is perfect. Your first dropper, you're gonna put in the detergent. Okay, that's your dish soap that you're using. And so you will do that, and then you're gonna prepare a shallow dish with very warm water. Okay, so what I did, just to give you a tip, I took the water and just so that you make sure you have enough water in there to cover your saliva. So you want it a little to go a little bit above that cup and also wherever your strawberry is. Okay. One thing I want to note that with this the strawberry one stayed in the shallow dish, but the one with the saliva started floating. So I took just a box of toothpicks to anchor it down, and that worked perfectly. Or anything heavy to anchor it down so that it doesn't float. You want it to stay in that shallow water and stay warm. You're going to do that for 10 minutes, okay? And so that's one of the tips that I have for you for that. And then the rest of it worked out perfectly. Okay, after 10 minutes, you let it cool to room temperature, and then you went to the next step, which is the cleaning method. With the cleaning, you want to use the coffee filter to strain your strawberry. You want the liquid. Okay, so what I did is I put the strawberry mixture into the filter and then I gently squeezed it. You don't want to squeeze it too hard because then it'll push the strawberry solid out and you don't want that. All you want is the liquid. And so you want to gently squeeze the filter or the paper towel that has the strawberry in it, gently squeeze it into an extra little cup. So all that you get in that cup is the liquid. Okay, so be careful. Don't squeeze it too hard. All right, so then you're putting in the contact solution or a tiny amount of the tenderizer, just a few drops. I added 4 drops, that worked out perfectly. And then you get a clean toothpick. So use 2 toothpicks for the procedure, 2 toothpicks for the cleaning. And when you get to this part, um, you're mixing it and you have another clean toothpick, so I guess 6. So 6 toothpicks is what you'll need. So you add a pinch of salt, okay, into the next step. You're going to gently mix it together, and then you're going to add enough of the chilled alcohol to fill the container. Okay, so chilling the alcohol. I'm going to tell you what I did. So I just took just a plastic cup, okay, and I measured out how much alcohol I needed. You need to be able to fill your little cups with alcohol. So I poured alcohol into the little cup and then dumped it into here, okay? And then filled it and dumped it into here. So I did that twice so that I could get enough alcohol to fill both containers. And then I put it in a container of ice, okay? so that it would chill. So what I did is I started chilling this before I started the experiment. Okay, so you want to do that first. So about time you get to the precipitation part, this is nice and cold and ready to go. Okay, so the first thing you want to do is you want to chill your alcohol at the beginning, and then once you've chilled it, once you get to the precipitation part, it's nice and chilled and ready to go. It should feel cold to the touch in the cup, okay? And then if you touch the alcohol itself, it should feel nice and cold, okay? So that's how you know if your alcohol is chilled enough. All right, so once you've gotten to here, then you're going to put in your chilled alcohol. And what this is going to do is it's going to precipitate the DNA. The DNA is going to look kind of cloudy in your solution. So if you have a cloudy substance in your solution, that's your DNA, okay? And so once you've done this, you're going to gently swirl your new toothpick that you have for this section and attempt to spool it around. Spooling it means you're going to stir it around in a circular motion and you're trying to get it to get your precipitate to me actually show you a toothpick. There we go. You want to get it to actually wind around your toothpick, okay? And so I'm going to show you what it looks like. Oh, that's hard to see. Okay, let me stop sharing so you can see that. Okay, so this is what it looks like for the strawberry, okay? So this is it. And so it's just going to kind of wrap around it and it looks like a blob. It looks like a little blobby substance on your toothpick. Okay. And so that's what that is your end result. That's what you're looking for. All right. So you're gonna take pictures of that. Make sure you get that 2 to 3 minute video on your lab, okay? Because you want to make sure that you are recording part of your experiment. 2 to 3 minutes, all we need. All right. If you have any questions, though, let me know. Enjoy the lab.